student. I am Mr. Vinod Thumre and we are studying plant physiology. The next topic from the today's lecture of plant physiology is fixation of carbon dioxide through Kelvin cycle. Now what is the Kelvin cycle? All photosynthetic eukaryotes, from the most primitive alga to the most advanced angiosperm, reduce carbon dioxide to carbohydrate via the same basic mechanism, the photosynthetic carbon reduction cycle, originally described for C3 species, the Kelvin cycle, or it is also called as reductive pentose phosphate cycle. It is the dark reaction. Okay, in previous lecture we discussed about the light reaction, that is the photosynthesis through the light reaction or the electron transport system, photosynthetic electron transport system. Here, a dark reaction, the location of dark reaction is stroma. All enzymes required for dark reaction, they are present in stroma, and that's why the Kelvin cycle which is occurred inside the stroma of chloroplast. Now the C3 cycle, the first stable compound is of 3 carbon, and hence the plant which obeys C3 cycle are called C3 plants. Now, what is the basic reaction? Carbon dioxide fixation, unbound carbon is converted into carbon fixed in carbohydrates in the bound form. Now, the Kelvin cycle, which proceeds in 3 stages. The first stage is carboxylation, the second stage is reduction, and the third stage is regeneration. In carboxylation, the CO2 acceptor ribulose 1,5-bisphosphate forming two molecules of 3-phosphoglycerate, the first stable intermediate of the Kelvin cycle. In reduction reaction, reduction of 3-phosphoglycerate forming glyceraldehyde 3-phosphate, a carbohydrate. In regeneration reaction of the carbon dioxide acceptor ribulose 1,5-bisphosphate from glyceraldehyde 3-phosphate. Here is the graphical representation of these 3 stages of Kelvin cycle, where in first stage ribulose 1,5-bisphosphate is converted into 3-phosphoglycerate by the reaction carboxylation. This is the start point of Kelvin cycle, where carbon dioxide is fixed by the important enzyme rubisco. In second stage, the 3-phosphoglycerate is reduced into the glyceraldehyde 3-phosphate. Here, the formation of glyceraldehyde 3-phosphate takes place. In this reaction, ATP and NADPH, they are required to carry out the intermediate steps to form glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is regenerated into ribulose 1,5-bisphosphate. Here is also the requirement of energy is required to carry out the intermediate steps in the Kelvin cycle to convert glyceraldehyde 3-phosphate into ribulose 1,5-bisphosphate. So basically, Kelvin cycle is operated in 3 steps. The primary stage is carboxylation, the second one is reduction, and the last stage is regeneration of RUBP, that is ribulose 1,5-bisphosphate. Let's see in detail the C3 cycle. The C3 cycle is carried out by in several steps. In previous we discussed that it is carried out in three basic steps. The first step is carboxylation, which is generally carried out by the enzyme which is called Rubisco. The substrate here utilized is ribulose 1,5 bisphosphate RUBP. For our convenience, we take a 3 molecule of RUBP and fix 3 CO2 molecules and again regenerate 3 RUBP molecule through whole C3 cycle. The RUBP, which is of 5 carbon compound, if we take 3 molecules, then the resulting product or the resulting molecules, they are of 15 carbon compound. 3 molecules we have take here, so the fixation of 3 carbon dioxide is takes place by the enzyme Rubisco and formation of 2 carboxy 3 keto arabinitol 1,5 bisphosphate is takes place. Here, due to fixation of 3 carbon dioxide and formation of 3 molecule, each RUBP is fixed with 1 CO2 molecule and hence the resultant product will be 3 molecules and each molecule is of 6 carbon and the total molecules they are of 18 carbon compounds. In next step, 2-carboxy 3-keto-arabinitol 1,5 bisphosphate is hydrolyzed into the 3-phosphate glyceric acid by the action of water and formation of 6 molecules of 3-phosphoglyceric acid is takes place. Here, these molecules are 3 carbon compounds and hence total 6 molecules they are formed and the total carbon number is 18 carbon. In next step, 3-phosphoglyceric acid is converted into 1,3-bisphosphoglyceric acid and the enzyme which carry out this reaction is called 3-phosphoglyceric acid kinase. Here the requirement of energy is fulfilled by the utilization of ATP molecules. Here for the one molecule there is requirement of 1 ATP. Hence here the 6 molecule they are converted into 1,3-bisphosphoglyceric acid. Hence the requirement of 6 ATPs are required and these ATPs are hydrolyzed into the 6 adenosine diphosphate. The resulting 1,3-bisphosphoglyceric acid is converted into glyceraldehyde 3-phosphate by the action of enzyme NADP glyceraldehyde 3-phosphate dehydrogenase. There is the enzyme which converts 6 molecules of NADPH into 6 molecules of NADP and the 6 inorganic pyrophosphates are liberated. For each molecule there is a requirement of 1 NADPH. Here 6 molecules they are converted into glyceraldehyde 3-phosphate, hence 6 NADPH are required to convert 1,3-bisphosphoglyceric acid into glyceraldehyde 3-phosphate. Total 6 molecules of glyceraldehyde phosphate they are formed in this reaction. Out of these 6 molecules, 1 molecule of glyceraldehyde 3-phosphate, 1 molecule of glyceraldehyde 3-phosphate is utilized either for starch synthesis or sucrose synthesis or it may transport to the cytoplasm. Remaining 5 molecules of glyceraldehyde 3-phosphate, out of that 5 molecule, 2 molecules of glyceraldehyde 3-phosphate, they are converted into the interconvertible form of glyceraldehyde 3-phosphate, that is dihydroxyacetone phosphate. Out of 5 molecules, 2 molecules of glyceraldehyde 3-phosphate, they are converted into dihydroxyacetone phosphate. So 2 molecule, each molecule is of 3 carbon compound, total 6 carbon. The remaining 3 molecules, they are as it is utilized for the regeneration of ribulose 1,5 bisphosphate. Now, out of out of 2 dihydroxyacetone phosphate molecules and 3 molecules of glyceraldehyde 3-phosphate, 1 dihydroxyacetone phosphate and 1 glyceraldehyde 3-phosphate, they combine together to form fructose 1,6 bisphosphate, and the reaction is carried out by the enzyme aldolase. Hence, both the compounds, they are of 3 carbon compound. The resulting product is with 6 carbon compound. Resulting product is with 6 carbon compound. Then this fructose 1,6 bisphosphate is further converted into fructose 6 phosphate. This fructose 1,6 bisphosphate is converted into fructose 6 phosphate, and the enzyme which catalyzes this reaction is fructose 1,6 bisphosphatase. Here, the fructose 6 phosphate and the remaining molecule of glyceraldehyde 3 phosphate, that is, out of 3 molecule, one is utilized for the formation of fructose 6 phosphate, and 2 molecules, they are still remaining. Out of that 2 molecules, one molecule is again utilized here and combined with fructose 6 phosphate to form erythrose 4 phosphate, and xylulose 5 phosphate. Both, they are formed from the condensation of fructose 6 phosphate and glyceraldehyde 3 phosphate. Fructose 6 phosphate is of 4 carbon molecule and glyceraldehyde 3 phosphate is 3 carbon molecule, total 9 carbon compound. They are derived from this reaction, and this reaction is catalyzed by the enzyme which is called transketolase. The resulting product is erythrose 4 phosphate, which is a 4 carbon compound, and xylulose 5 phosphate, which is 5 carbon compound. Then the erythrose 4 phosphate, here the erythrose 4 phosphate is combined with the remaining molecule of dihydroxyacetone phosphate. We know that previously we discussed 2 molecules of dihydroxyacetone phosphate, they are converted from glyceraldehyde 3 phosphate. Out of that, one is utilized here and the remaining one is utilized here. Erythrose 4 phosphate is combined with dihydroxyacetone phosphate to form pseudoheptulose 1,7 bisphosphate. Pseudoheptulose 1,7 bisphosphate, which is a 7 carbon compound, because erythrose 4 phosphate is a 4 carbon molecule and dihydroxyacetone phosphate is 3 carbon molecule. Hence the resultant product is with 7 carbon compound, and the reaction catalyzed by the enzyme is called aldolase. Here is the enzyme aldolase. Then pseudoheptulose 1,7 bisphosphate is converted into pseudoheptulose 7 phosphate. Here the phosphate group from the 1 carbon position is removed by the enzyme pseudoheptulose 1,7 bisphosphatase. Pseudoheptulose 1,7 bisphosphatase. Then The pseudoheptulose 7-phosphate is condensed with the last remaining molecule of glyceraldehyde 3-phosphate. The last remaining molecule of glyceraldehyde 3-phosphate is condensed with pseudoheptulose 7-phosphate. Now, the pseudoheptulose 7-phosphate is a 7-carbon product and glyceraldehyde 3-phosphate is 3-carbon product. From those two products, the one molecule of ribose 5-phosphate, one molecule of ribose 5-phosphate and one molecule of xylulose 5-phosphate are forms, and the reaction is catalyzed by the enzyme which is called transketolase. Here the xylulose 5-phosphate, the one molecule which is derived from the condensation of glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate, and the remaining molecule is the again one molecule which is synthesized here, xylulose 5-phosphate. Hence one molecule which is derived here with the combination of fructose 6-phosphate and glyceraldehyde 3-phosphate, and the another molecule which is derived from pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. So here the total 2 xylulose molecules are formed. 2 xylulose molecules are formed. 2 xylulose molecule. Here the one ribulose 5-phosphate is converted into one ribulose 5-phosphate, one ribose 5-phosphate is converted into ribulose 5-phosphate. So the one molecule of ribulose 5-phosphate derived here, and the remaining two molecules, that is the xylulose 5-phosphate, they are also converted into ribose 5-phosphate. They are also converted into ribulose 5-phosphate. So one molecule from the conversion of ribose 5-phosphate and two molecules of xylulose 5-phosphate, they are converted into ribulose 5-phosphate. So total 3 ribulose 5-phosphate they are derived. The reaction ribose 5-phosphate is converted into ribulose 5-phosphate is carried out by the enzyme isomerase, and the reaction xylulose 5-phosphate is converted into ribulose 5-phosphate is carried out by the enzyme which is called ribulose 5-phosphate epimerase. So total how many compound generated here? The 3 ribulose 5-phosphate they are generated here. Each molecule is of 5-carbon compound. 3 molecules they are prepared here, and they converted into the end product of this Kelvin cycle, ribulose 1,5-bisphosphate. Ribulose 1,5-bisphosphate, and the enzyme which carry out this reaction is ribulose 5-phosphate kinase. Here 3 molecules they are converted into ribulose 1,5-bisphosphate. In that reaction, the additional phosphate at the position 1 is added by the hydrolysis of ATP to ADP. Here 3 molecules they are hydrolyzed, 3 molecules they are converted into ribulose 1,5-bisphosphate. So 3 ATPs they are required. So this is the Kelvin cycle which starts from RUBP to the end product RUBP which is regenerated. So the first step is carboxylation, the second step is reduction, and the third step in this Kelvin cycle is regeneration. Those are the reactions which are seen before in previous slide. Thank you.